welcome to this class on excitation and inhibition within the spinal cord, part 2. In this class we will be talking about 1A interneurons and its function. So, it is written with Roman numeral 1, but for clarity in this class I am going to call this as 1A with numeral 1 and A, because otherwise some students read it as IA interneurons. So, that is actually 1A interneurons, and the feedback loops that are involved by 1A interneurons, and those that involve a combination of 1A interneurons and Renshaw cells. We saw the case of Renshaw cells yesterday. Let us remind ourselves what it is. So, Renshaw cells are activated by motor neurons and they inhibit the motor neuronal pool that excites it. A form of negative feedback is what we said, phenomenon also called as recurrent inhibition. Okay. There is more to that. This is just one part of a relatively larger feedback loop. We will see the remaining parts, and we saw that the Renshaw cell is an inhibitory interneuron, but there is one more inhibitory interneuron which we will call as 1A interneuron. So, pronounced 1A, okay. But usually in textbooks it is written like that, it is not IA interneuron, but rather 1A interneuron. Okay. These are excited by the proprioceptor neurons of the other muscle, of the agonist muscle. Let us consider a simple case. Here is one joint. So, that is one joint that is spanned by only two muscles. Let us consider a theoretical case. There are only two muscles, one is the agonist, the other is the antagonist for the purpose of this discussion. Let us suppose that this antagonist muscle is contracting or is trying to reduce that angle theta. That angle is trying to reduce. When that happens, what will happen is that because this muscle is connected to both bones like that and this muscle is connected to both bones like this. Because of this reason, as this muscle shortens, this muscle is going to reduce size. As this muscle is going to shorten, this muscle is going to be stretched, is going to be elongated. When this muscle is elongated, we saw earlier a case of a length sensor, which are muscle spindles, right. So, these are of two types, primary spindle and secondary spindle, or 1A afferent, 1A afferent or 2 afferent, is it not. 1A we saw is responsible for detection of both length and velocity, 2 is responsible just for the detection of length. 1A can, depending on the sensitivity of the dynamic and static gamma motor neuron, depending on that it will act either as a length sensor or as a velocity sensor. This is what we saw. So, when this muscle, when the antagonist is contracting, the agonist will stretch and then the sensor will start firing, will start sending an input that is proportional to the amount of stretch, is it not. So, it is a length sensor. So, as soon as the length becomes increased, this is going to start sending information. So, the sensory part of this proprioceptor neuron is here intertwined with the intrafusal fibers, actually in the center, right, intertwined with the intrafusal fiber here like that. Cell body or the soma is in the spinal ganglia, and we said it has a characteristic T shaped axon, right, like that. So, conduction happens antidromically in that direction towards that, there is a little bit of processing, very little bit of processing, and then orthodromically in that direction. Then what happens? So, this proprioceptor neuron is exciting an inhibitory interneuron which is called 1A interneuron. So, this 1A interneuron is activated. What it will do is it will inhibit. So, here the open circles are excitatory, those that are filled, empty circles are excitatory, those that are filled are inhibitory. Okay. So, this 1A interneuron inhibits the antagonist motor neuronal pool. This antagonist motor neuronal pool is the one that is activating and causing contraction. So, in a way, as soon as it is realized that this muscle is stretching, that the agonist muscle is stretching, the proprioceptive information goes via 1A interneuron and inhibits the motor neuronal pool that and prevents contraction of this muscle. What will then happen? Then this contraction will reduce, right. So, the stretching will reduce, a way of a method in which I am implementing negative feedback. Let us assume I am the agonist muscle, somebody is pulling me, the antagonist muscle is pulling me, I am trying to ensure or minimize the amount of that antagonist activation. So, I will not be pulled too much. So, that information goes via the proprioceptor neuron through the 1A interneuron. Note, the information from the proprioceptor neuron is always excitatory. This is always excitatory. The information from the proprioceptor neuron is always excitatory. As the length increases, the excitation amount is going to increase, the rate of firing is going to increase. This is true for all proprioceptor neurons. In this case, this is 1A afferent. This is true of 2 afferents, this is true of 1B. What is 1B? 1B is the afferent associated with Golgi tendon organs, a different case, does not matter. All the proprioceptor neurons are excitatory. So, this the probability that the 1A interneuron is going to increase its firing is increased when proprioceptor neuron sends an excitatory command to this 1A interneuron. As soon as this is getting excited, however, this 1A interneuron inhibits, sends inhibitory neurotransmitters, inhibits this alpha motor neuronal pool, thus preventing any further contraction, or at least to some extent it is reducing the probability that the contraction is going to continue sustained or keep on increasing. Why is this needed? Otherwise, this muscle can keep on contracting, and otherwise the antagonist muscle can keep on contracting and the agonist muscle can keep on stretching. At some point it could cause damage, or to create a balance in the amount of contraction and stretching between these two muscles. Also, we are not just interested in achieving a particular posture, which is theta. So, when I am saying that is the theta I want, it is not just that we are interested in achieving that particular theta, I am also interested in achieving that with a relative sense of ease or smoothness. Humans like this. So, we like to achieve this with some smoothness. So, to ensure that there is some amount of moment to moment control, at least to some extent, there are some delays associated with this. Obviously, there is some delay here, there is some delay here, there is some delay here and some delay here. So, there is definitely some delay, but despite those delays there is some amount of control that is exerted on the antagonist muscles alpha motor neurons to ensure that the probability that they will contract is at least slightly reduced due to this. Also, one thing to note is that it is not necessary that the agonist muscle will stretch only when the antagonist muscle is contracting. It is also possible that the agonist muscle will stretch due to an external load. An external load could cause stretching, even in those cases, because it is not like the proprioceptor neuron can know who is causing the stretch. The proprioceptor neuron just detects the amount of stretch. So, as soon as the stretch comes, so it sends the excitatory information to 1A interneuron, which further sends an inhibitory information to the antagonist motor neuronal pool. So, regardless of what the source of stretching is, the antagonist motor neuronal pool will get inhibited to some extent. Also, we must realize one more thing. What is not shown in this picture, just to prevent the complications that might arise, is the following. Let us suppose the opposite is happening. Okay. Let us suppose that it is the agonist muscle that is contracting. In other words, that muscle is reducing in its length, that muscle is getting stretched. Then what will happen? As soon as this muscle is getting stretched, what is not shown in this picture, now I will draw, is that there is a proprioceptor axon here whose cell body is lying in the spinal ganglion, let us say that is the spinal ganglion, and there is a T shaped axon having both orthodromic and antidromic conduction, and there is one more neuron here. Okay. This is the 1A interneuron. So, this is exciting a different 1A interneuron, and that 1A interneuron is going to inhibit this alpha motor neuron, that alpha motor neuron. Why? That alpha motor neuron is the one, or that pool of alpha motor neurons is the one that is causing this contraction. I want to prevent or minimize or reduce slightly to some extent or control the amount of contraction that is happening in this alpha motor neuronal pool. So, so there is reciprocal inhibition. So, in other words, the projection is going to the opposite muscle. So, this is true for all muscles, this is true for all the muscles that have counterparts. So, in every case there is somebody else. So, in this case, so when this is getting, when this antagonist is getting stretched, it is going to excite this one, there is a different 1A interneuron here, that 1A interneuron is going to be excited, and that guy is going to come, or that 1A interneuron is going to inhibit the alpha motor neuronal pool that is causing the contraction of this muscle. So, this kind of projections that go to the other muscle or afferent projections that go to neurons that innervate other muscles are called as heterogenic projections. Those projections that go to the neurons innervating the same muscle are called as autogenic projections. Okay. In this case, these projections are heterogenic, and when the agonist muscle is getting contracted, the antagonist will inhibit the agonist motor neuronal pool. When the antagonist muscle is getting contracted, the agonist will inhibit the motor neuronal pool of the antagonist, or more simply, because this terminology is a little confusing, we can use biceps and triceps. If biceps is contracting, then triceps will get stretched, then triceps will inhibit the motor neuronal pool that innervates biceps, because it is that motor neuronal pool that is causing this contraction. So, I want to minimize that. Or if triceps is contracting, then biceps will get stretched and it will send inhibitory information through 1A interneuron to reduce, minimize or balance the amount of contraction in the triceps. So, I do not want the triceps to keep contracting forever. So, I want to minimize it. So, this kind, so there is this balance that is achieved between agonist and antagonist. So, there is inhibition reciprocally between these two cases. Okay. Now, we have Renshaw cell is activated by alpha motor neuron, right. This is what we saw in the previous class, is what we saw in the previous slide. So, whenever this alpha motor neuron is firing, it is going to activate this Renshaw cell. What this Renshaw cell does, one part of what we saw yesterday was that it inhibits the same alpha and gamma motor neuronal pool for that agonist and its agonist. So, for that muscle and its agonist, all the muscles that perform approximately the same function. So, it is going to not just inhibit one muscle, it may also inhibit other muscles, it will also inhibit not just alpha motor neuron, it will also inhibit gamma motor neurons. This is the function of this is one function of Renshaw cell. One other function that it does is it also sends an inhibitory connection innervation to 1A interneuron that innervates the antagonist. Let us try to analyze this situation. What this does is, as soon as this alpha motor neuron sends a command, the Renshaw cell becomes activated or gets excited and sends inhibitory command to 1A interneuron. So, what will that do? That let us try to analyze what will be the action of the alpha motor neuron itself. This alpha motor neuron, when it is firing, will cause a contraction of this muscle as has been shown previously. It will cause a contraction of that muscle. It will cause a contraction which will cause a stretching in this muscle, is it not? This is what this alpha motor neuronal activation will do. In a way, if I inhibit this 1A interneuron, if I inhibit this 1A interneuron is already inhibiting this antagonist pull. In other words, there is one minus here, there is one more minus here. So, effectively what I am doing is I am increasing the probability that an excitatory command is going to be sent to this antagonist muscle, or in other words, there is a contraction here. I want to increase the probability that the antagonist, there is a contraction in the agonist, I want to increase the probability that the antagonist will also contract, so as to balance the contraction provided by the agonist. So, as soon as this agonist is contracting, I want to contract the antagonist, or at least increase the probability that it will contract, right. That is going to happen only if I excite this alpha motor neuronal pool. This is the alpha motor neuronal pool, is it not? This I want to excite, but what the Renshaw cell is doing is it inhibits the 1A interneuron which is inhibiting, it inhibits the 1A interneuron which is inhibiting the alpha motor neuronal pool, or in other words, it is increasing slightly the probability that this alpha motor neuronal pool is going to reach threshold or is going to get activated and going to contract, thus providing one more level of negative feedback. So, there are at least 2-3 levels that we have, levels of negative feedback that we have seen. One is this itself, this loop, is it not? So, the alpha motor neuronal pool excites and there is negative feedback here. The alpha motor neuronal pool excites the Renshaw cell and the Renshaw cell inhibits the alpha motor neuronal pool, that is one level. 2, the muscle, the agonist muscle contracts or the antagonist muscle contracts, that causes a stretching of this muscle, the agonist muscle, which is inhibiting, which inhibits the contraction of the antagonist through the 1A interneuron, that is that loop, is it not? That is the bigger loop. Then there is the loop that covers that. So, note in this case the first two are cases of inhibition against an excitation. Whenever there is an excitation, there is going to be an inhibition. So, excitation happens in the Renshaw cell, inhibition happens, inhibition is provided by the Renshaw cell, that is first. Second is excitation happens in this alpha motor neuronal pool, inhibition is provided by the 1A interneuron through the mediated obviously by the proprioceptor neuron. These are two. The third is a different case in which an excitation is provided or an inhibition to inhibition, or that and that. These two are provided through the activation of Renshaw cell. As soon as this alpha motor neuron is getting excited, it is exciting, not inhibiting, it is inhibiting the inhibition of the other opponent, other pole, or so it is a very fine design. So, depending on the case, inhibition or excitation could happen, and in this particular case an inhibition of inhibition or a disinhibition happens. Very important terminology, this disinhibition. We will discuss this in great detail when we discuss brain structure, especially basal ganglia. This terminology disinhibition is performed at this is happening in the spinal cord, but this also happens at several levels in the brain, especially in the basal ganglia. We will discuss this in greater detail later. So, what have we seen? Let us remind ourselves, this is a schematic of what is going on. What is going on is this. So, suppose the agonist is contracting and producing a force, right, that causes, that increases the length. So, the agonist is contracting, its length is reducing, then the antagonist is getting stretched. When the antagonist is getting stretched, what will happen? The 1A afferents will become active and they will send excitatory commands to 1A interneurons, which will inhibit the alpha motor neurons of the agonist. So, that is the negative feedback loop that we are talking about. As soon as this alpha motor neuron fires, then there is a balance that is brought about by the action of 1A interneuron sending this negative information. This is the first negative feedback loop that you see. Then what else? Then we saw the case where the alpha motor neuronal pool activates the Renshaw cell, which further inhibits the same alpha motor neuronal pool, that loop, that is the second feedback loop. Then what we also saw was that this Renshaw cell not only inhibits the alpha motor neuronal pool, but also inhibits the 1A interneuron of the antagonist. This is which 1A interneuron? This is antagonist 1A interneuron. This antagonist 1A interneuron is inhibiting the alpha motor neuronal pool of the antagonist of this. This is the alpha motor neuronal pool of the antagonist that is getting inhibited by the 1A interneuron. Its activity itself is inhibited. So, that is minus into minus plus, in other words effectively increasing the probability that this alpha motor neuronal pool will fire or increasing that probability. Okay. So, what we have seen is there is one more loop that involves that. In this case, this is negative because this is this might appear as plus, minus into minus is plus, is what it would appear as. This is negative because it is activating the antagonist, in that sense it is negative. Okay. So, one more level of hierarchy in which negative feedback appears in this. So, balance in this system appears or manifests at multiple levels. By the way, we are not done, there is more to this story. With this, at least we are able to realize that there are 2 or 3 levels at which it is happening within the spinal cord. Then there are other levels of spinal hierarchy. Actually, there are other things within the spinal cord, that is 1B information that comes from Golgi tendon organs. It is not like it is unused, it must be used somewhere. How is it used? And other things that are not discussed so far. So, we will discuss all those things and other such loops. So, we will, this is going to be a recurring theme in this course. So, this is going to come several times, from time to time I am going to say, so this neuron is inhibiting, so this neuron is disinhibiting. By the way, it might appear like what is this disinhibition? Inhibition of inhibition is not actually excitation. There is a difference between exciting somebody and not inhibiting somebody. There is a difference between these two, is it not? So, it is probabilistic, the probability that this is going to fire, at least we should agree that the probability that a neuron is going to fire is increased when it is disinhibited, when the inhibition on it is reduced. So, the probability that you are all going to perform well or or reach great heights is going to be increased if the inhibitions on you is removed, something of that sort. But yet there must be other sources, or there may be other sources which may be spontaneous or control that may cause excitation, but at least we are preventing, we are removing the obstacle to excitation in that sense. So, it is not exactly excitation, but it can be considered to be a form of excitation, or at least we are increasing the probability it is going to be excited. Or consider that if inhibition is present, then the chance that it is going to become excited is at least lower in the presence of inhibition versus in the presence of disinhibition. In that sense it can be considered to be an excitation. So, what we have seen in this class is the case of 1A interneuron, its important function in inhibiting the antagonist alpha motor neuronal pool when the agonist muscle is contracted, when the agonist muscle is stretched. When the agonist muscle is stretched, the antagonist alpha motor neuronal pool is inhibited, and we said that remember that this inhibition is going to happen regardless of what causes the stretching of this muscle. The stretching can happen because of the contraction of the antagonist, or it can happen because of an external load. Either way, the alpha motor neuronal pool of the antagonist will get inhibited, because 1A interneuron is just performing its function, 1A afferent is just performing its function. What is the function of 1A afferent? As soon as the length is increased, fire and tell 1A interneuron to inhibit the antagonist alpha motor neuronal pole. That is the function. They do not know what is causing this stretch, that is a more fine information. And feedback loops concerning 1A interneuron, there is one that involves reciprocal inhibition between two muscles, for example, and this exists throughout the body in multiple muscles. Okay. And then feedback loops involving Renshaw cells, the one that involves a smaller feedback loop that involves so there are two of these, one involves direct inhibition of the same alpha motor neuronal pool, that is what we saw, alpha motor neuronal pool and gamma motor neuronal pool, just the motor neuronal pool. And within the Renshaw cells there is one more, that it in disinhibition or excitation of the antagonist motor neuronal pool. So, by this, because I am activating the opponent, because I am activating the antagonist, negative feedback is achieved. So, we have seen these three cases, we will see more cases and those that involve reflexes and stuff from future classes, in future classes. So, with this we come to the end of this class. Thank you very much.